Okay, yeah. Thank you very much, Mansour. It's a great pleasure to be here. Yeah, I'll just briefly introduce myself. I think some of you, most of you know me from the last couple of small meetings that we have. So my lab is interested in biophysical aspects related to polymer encapsulated nanodiscs. And more recently, we've also become interested in developing, synthesizing, validating new polymers. And that's actually the topic of today's talk. So I keep it relatively short. I really took the name of this seminar series, Journal Club, very literally. So what I'm going to show you is unpublished, so that's very fresh data. So don't expect a polished talk. This is really going to be something like a journal club. And I try to keep it short so we have more time for discussion, and I skip a formal introduction because I suppose that all of you know what amphiphilic copolymers are and what they're being used for in the small community. So the goal of this project was to develop new polymers that extract membrane proteins and lipids very efficiently. Those nanodiscs should be small and narrowly distributed, so they are amenable to cryo-electron microscopy. They should also be amenable to other downstream analysis and manipulation methods like microfluidics, for instance. The polymers should be we should be able to produce those polymers in large amounts, right? We don't want to limit ourselves to milligram amounts of polymers, but we want to have polymers that can be used by more labs in the community. And we wanted to avoid IP issues because that's something that's always annoying, particularly when it comes to SMA. So this intro sounds very much like the intro I gave a couple of weeks back at the last small web meeting where we introduced glycodipmer. The major difference, or the new element now, is that these polymers should now be completely electroneutral. So with glycodipmer, we have reduced the charge density on the polymer because some of the carboxylate groups were derivatized with the sugar moiety. Now in this case, we aimed at completely removing the electric charge on the polymers. And the two polymers that I will introduce to you now is shown here. So on the left, you see what we call SB-DIPMA, so SB stands for sulfobetaine because this moiety is the sulfobetaine group. So as you can see here, we have the DIPMA backbone. We started from the anhydride form of DIPMA, and then we turned this anhydride into a Malay imide. And this Malay imide has a quaternary ammonium group, which is essentially always positively charged here, and it has a sulfonate group, which is under physiological pH values always negatively charged. So it's vitrionic but net neutral. And the second polymer that you see on the right-hand side is SB-SMA, so sulfobetaine SMA. In this case, you see that there is a slight modification in comparison with SB-DIPMA. With SB-DIPMA, we use the C3 link here. With SMA 2:1, we use the C2 link. And the reason for this is that DIPMA is intrinsically less hydrophobic than SMA, so we thought. So what I was telling you about with these two structures, so on the left you see the polymer that we call SB-DIPMA, where SB stands for sulfobetaine, so this is a sulfobetaine group, and on the right you see SB-SMA, so this is made from SMA 2:1, this is made from DIPMA. So in both cases, we converted the anhydride versions of those polymers into Malay imide, and we coupled the sulfobetaine group. The only difference between the two pendant groups is that in the case of SMA, we use the C2 linky, whereas in the case of DIPMA, we use the C3 linky because DIPMA is intrinsically less hydrophobic than SMA. So we thought it would be a good idea to render it a little more hydrophobic by using a longer linky. And I mean, the first thing we did was to look at the solubilization of lipid vesicles. So here on the left-hand side, you see intensity-weighted particle size distributions that we got from dynamic light scattering. In gray, you see the size distribution of vesicles that had not been exposed to any polymer. And here in orange, red, and pink, you see 3 traces for increasing Sp digma to lipid mass ratios. So what you see is that with increasing polymer to lipid mass ratio, the size decreases as expected, but in all 3 cases, the size is around 10 nanometers, so as you would expect it for nanodiscs. On the right-hand side, you see a comparison of digma versus Sp digma. So one of the issues with digma is that it forms relatively large nanodiscs. So even in the presence of excess polymer, you typically get nanodiscs on the order of 20 or more nanometers. So this is not the case with Sp digma. You see here that first of all, Sp digma is more efficient than digma, so the size decreases from a value typical of vesicles to a size typical of nanodiscs at a lower polymer to lipid ratio. And you also see that the size that those nanodiscs have is narrowly distributed around 10 nanometers, right? We looked at the same process using phosphorus NMR spectroscopy. I think you can keep this very short because it's the same stuff that I always show in my talks. So here you have a bunch of NMR spectra where we look at the appearance of an isotropic peak, which reflects the presence of nanodiscs. So in the absence of polymer, you have no peak because you don't see the vesicles. In the presence of increasing concentrations of polymer, you see an increase in this isotropic peak. If you do that at different concentrations of lipid, like 2 to 8 milligrams per milliliter DMPC in this case, you get different titrations, and from a series of such titrations, you can then construct a so-called phase diagram in which you have the lipid concentration on the x-axis and the polymer concentration on the y-axis. And this phase diagram essentially tells you how much polymer it takes in order to solubilize a given amount of phospholipid. So on the next slide, you see 2 very different kinds of data. In the upper left corner, you see a negative stain electron micrograph. And here you see on the one hand, like a face-on or a top-down view of some nanodiscs. So for instance, here, those red arrows pointed nanodiscs that were like photographed, so to say, from above or from below, if you will, whereas the black arrows point at rouleaus, so it's stacks of nanodiscs. And this is an artifact that comes from the staining procedure, so this is something that you would not expect and that you don't see in solution. But this is induced by uranyl acetate, but it's a nice artifact because it really helps us visualize those nanodiscs in side-on view. So you can really look at the thickness or at the height of individual nanodiscs in such a stack to confirm that what you have in those nanodiscs is indeed a lipid bilayer. Here in the lower right corner, you have zeta potentials of different nanodiscs and different lipid-free polymers. So the gray line here indicates the zeta potential of DMPC. So PC, phosphatidylcholine, is a switch ionic phospholipid, but because there is differential absorption of counterions to those phosphocholine moieties, they still have a small but non-zero zeta potential. So this is typically on the order of -2 millivolts. And now you see in comparison with the DMPC, digma, so pure digma without any lipid, or digma nanodiscs, of course, give rise to strongly negative zeta potential, which is of course due to the many carboxylates on the polymer. And in contrast with this, Sp digma, both in isolation or in combination with lipids, so in the form of nanodiscs, gives rise to a zeta potential that is very close to that of polymer-free DMPC. So, I mean, these data show that in this case, we have really succeeded in abolishing the net charge on the polymer. This has some nice consequences when it comes to the influence of pH or the effect of magnesium or also calcium. So first of all, you see that these polymers do not show a strong pH dependence anymore, at least not around neutral pH. We also tried low pH values and still didn't see any significant changes. But I guess for most applications, something like pH 5 or pH 3 is not really relevant. But around neutral pH, there is really no difference in the efficiency of those polymers. And in terms of magnesium, also calcium, or even trivalent cation concentrations, you can go up to 70, 80, or even higher millimolar concentrations without seeing any precipitation of the polymer or of the nanodiscs. This slide shows a summary of some DSC data. So this is differential scanning calorimetry, which is a very sensitive method to look at the thermotropic phase transition, or the thermotropic behavior of lipids in nanodiscs. So again, the gray trace here shows the thermotropic phase behavior of DMPC, so fully saturated phospholipid in polymer-free vesicles, and you get a sharp peak at around 24 degrees Celsius. And then you see two traces for DIPMA nanodiscs and for SB DIPMA nanodiscs. So in both cases, you see that the peak is now much broader, which is simply due to the fact that you have just a few lipid molecules in one nanodisc, whereas in the vesicle you have some 10,000 or some 100,000 lipid molecules. So this is one thing. And the second thing that's more interesting for us to look at is the change in the maximum, so the temperature at which this CP curve goes through a maximum. And this is plotted more systematically on the right-hand side for DIPMA. That's this trace here, which has a peak and then decreases a bit, and also SB DIPMA, which behaves more smoothly. So basically the take-home message from this slide is that SB DIPMA has a very mild impact on the thermotropic phase behavior of DMPC, as is also the case for DIPMA actually, but still for us it is important to show that SB DIPMA is not aggressive, because this is usually what happens when you render a polymer more efficient in terms of lipid solubilization efficiency. It also becomes more aggressive in the sense that it abolishes the phase transition, which shifts the phase transition to much lower temperatures. So to sum this up, I mean, all this in vitro stuff so far, we have two polymers that have zero net charge. They're resistant to changes in pH and also to the presence of di and trivalent cations. They efficiently form nanodiscs that are well defined in terms of size and size distribution, and they have very gentle effects on the encapsulated lipids. Now the big question is, of course, what does this mean for protein extraction? And here I have a slide that looks a bit busy now because I cannot use any animations. So we should start here in the upper right corner. What you see here is a protein extraction experiment that we did on HeLa, on human cells, on HeLa cells, at different polymer concentrations. So what we did was to add different concentrations of SB DIPMA to the supernatant of a HeLa cell culture, and then we didn't break up the cells, we basically just decanted the supernatant and used the SDS page to look at the protein composition of the supernatant. And in the absence of polymer, you don't really find much as expected, but increasing the polymer concentration up to like 14 milligrams per milliliter, which corresponds to 1.4 weight%, you see that there are many different bands showing up in this lane, which tells us that lots of different proteins are becoming extracted with increasing concentration of SB DIPMA. Then in the upper left corner, you see a quantitative comparison of SB DIPMA versus DDM, which is kind of the gold standard detergent. So you see here that SB DIPMA is not quite as efficient as DDM, but it's also not too bad, right? I mean, it achieves about 50% of solubilization efficiency in comparison with DDM, and it achieves its maximum efficiency at relatively low polymer concentration of around 4 milligrams per milliliter, which corresponds to 0.4 weight%. And the major advantage of SB DIPMA in comparison with unmodified DIPMA is that the nanodiscs that are formed upon application of the polymer to the cell culture are much more homogeneous than those you get from treatment with DIPMA. So here you see elution profiles from size exclusion chromatography. In red, you have two different traces taken at 240 nanometers and 280 nanometers, so 240 nanometers, mainly reflects the concentration of the polymer, whereas the 280 nanometer trace reports on the protein concentration. And those gray bars that you see in the background, the total protein concentrations that are determined independently using a BCA assay. And you see that for SB-DEBMA, this peak, even though it's still broad, right? Because it's a very crude mixture of thousands of different proteins. So even though this peak is still broad, it's reasonably homogeneous, and the two spectroscopic signals overlap quite nicely. Whereas in the case of DEBMA, you have a much broad distribution of nanodisc sizes. You also have quite a bit of protein that is lost in the very first fraction that is simply flushed through the column without really interacting with the column material. So basically what you see here is that the SB-DEBMA or SB-DEBMA nanodiscs are more readily amenable to this kind of assay than DEBMA nanodiscs are. And we then used mass spectrometry to look at each individual fraction that was eluded from during the SEC experiment. And here I'm just showing you 4 examples of proteins that either elude on their own, like VDAC, for instance, or PAW. So VDAC is an abundant beta barrel membrane protein from the inner mitochondrial membrane, and this protein has been shown to exist as monomer, as a dimer, or as a trimer, which also gives rise to a relatively broad elution profile. The more interesting proteins are shown on the left-hand side. So here you have, for instance, 2 subunits of ATP synthase, which also comes from the inner mitochondrial membrane. So A1 is the alpha subunit and B1 is the beta subunit. And these 2 proteins have very different sizes, but they co-elude perfectly with one another, which is also a very strong indication that this complex is still intact. The same is true here for the insulin receptor and its binding protein. Those 2 also form a tight complex that can be extracted with the aid of SB-DEBMA. Okay, another application that I want to show you is MDS, microfluidic diffusion sizing. I would have a short animation here. I'm not sure if it runs now. If you have attended the last small web meeting, you know this already. If not, I'd like to explain it briefly to you. So this is a microfluidic technique that is based on mixing one channel in which you have your analyte, which means your protein of interest, with a second channel which contains just buffer or water. And since this is microfluidics based, there are no turbulences, everything is laminar. So the only way for a molecule to get from the upper channel to the lower channel is by diffusion. And you then separate those 2 channels and you determine the protein concentrations in the original channel, in the cis channel, so to say, and in the other channel, the trans channel. And from the ratio of protein concentrations in the undiffused and the diffused fractions, you can calculate the diffusion coefficient, and from this you can make conclusions as regards the size of the protein. In our case, we use this to look at protein or peptide membrane interactions. And this is now again a very busy slide, and we should again start here in the upper right corner. The first example that we looked at was a protein called GB1. This is not a membrane protein. It should also not interact with membranes. So this is our negative control. And we see that in the absence of lipids, this protein has a hydrodynamic diameter of around 6 nanometers. As we added the MPC or the MPG, the MPC mixture in the form of SB-SMA nanodiscs in this case, we didn't see a significant change in the hydrodynamic diameter. So again, this is a negative control. We expected to see no change and we didn't see any change. Then the more interesting case is shown here in the upper left corner. This is alpha-synuclein. And alpha-synuclein is soluble but membrane interacting peptide that is known to have a strong preference for anionic phospholipids. So here we expected to see no interaction with DMPC, so with switch ionic lipid, but a strong interaction with mixed DMPG, DMPC membranes. And again, this is what we observed. So here in the absence of lipids, we got the radius or the diameter of roughly 7 nanometers. This did not change as we added the MPC in the form of SB-SMA nanodiscs. and it increased drastically when we added DMPG plus DMPC encapsulated by SB SMA. And in the lower half of the slide, you see a similar experiment that was done on ACTH. So this is a peptide hormone. And here you see a clear advantage of SB, DEBMA, or SB SMA over unmodified SMA. So the problem with this peptide is that it is polycationic, which means that it tends to interact non-specifically with polyanionic polymers. So this peptide, you would not expect to see any interaction with DMPC, and this is indeed the case for the MPC encapsulated by SB SMA or SB DEBMA, but you see strong and unspecific interaction when you use DMPC that is encapsulated by SMA 2-1. And this is due to unspecific interactions of the peptide with SMA. And in contrast with that, as soon as you use an anionic phospholipid like DMPG, you also see strong binding with SB SMA and SB DEBMA. And here you have the same take-home message again, now plotted in a somewhat different way. So on the x-axis, you have the lipid to peptide ratio on a logarithmic scale. So basically, because this was the titration, you would expect the sigmoidal transition in such a plot, and this is indeed what you see for DMPG, DMPC mixtures, whereas for DMPC only, you don't see any change in the hydrodynamic diameter. Okay, and the last kind of experiment that I'd like to show you here is cell-free protein synthesis. I mean, that's also something that people have tried in the past, but as far as I know, nobody has succeeded with SMA or DEBMA, because the point is that the in vitro translation machinery, of course, requires magnesium, and getting the magnesium concentration right in the presence of polyanionic polymer always tends to be a headache. So what you see on the left-hand side is a control experiment in which we tried GFP. So there's no membrane protein involved in this case. We just looked at the effects of those polymers on the in vitro synthesis of GFP, of green fluorescent protein. And here in gray, you see the GFP control, which means in the absence of any polymers. Here you have the results, or the, let's say, the non-results that you get in the presence of DEBMA and in the presence of SMA 2-1. And you see that those two polymers essentially completely suppress protein synthesis. And with SB DEBMA and SB SMA, you get decent yields. Yeah? It's not exactly the same as in the absence of polymer, but you get about 50% of the maximum yield that you would expect in the absence of polymer. And in the lower right corner, you now see data on a membrane protein. So this is proteurhodopsin in the presence of increasing concentrations of either SB DEBMA or SB SMA. And here you see that there is a strong dependence on the polymer concentration. So if the polymer concentration is too low, the yields begin very low, but increasing the polymer concentrations to 1.6, 2.6 mg per ml allows pretty good yields. And again, I mean, 1.6 milligram per milliliter means 0.16%. Yeah? I mean, just to put this in perspective, with the usual 2.5% that many labs are using. Okay, so this is it, ready for the formal part of my talk. As I said in the beginning, the goal here is to develop polymers that extract proteins and lipids with high efficiency, that form small nanodiscs that remain of the downstream analysis and manipulation. And also those polymers should be producible in large amounts and at decent cost, because we'd like to make them available for the entire small and deep mold community, and they are electroneutral. Of course, these are not the first polymers that are electroneutral. So there are a number of polymers that have been shown to be electroneutral, but most of them either do not extract proteins directly from membranes, so they're not soluble in the pH range, and which they are electroneutral. I think the only exception is ZSMA, but the issue with ZSMA is that it has to be synthesized de novo, and this renders it quite expensive. So yeah, as always, I thank our funding agencies and collaborators, and I'd like to stop here and would be happy to take your questions. And thanks again, Mansour, for inviting me to this Friday afternoon, a Friday morning seminar for you.
Thank you so much. Very exciting. Yeah, feel free to ask your questions, everyone. We are just few people. Any questions? I have one actually. For example, the formulation of these electroneutral polymers. Have you, if you go back to the slide, that's you had one sulfitryl group, I would say. No, it's not it's sulfate group. Did you
It's a
sulfate? Yeah.
sulfate.
Yeah. Did you replace it with something like carboxylic acid? Something. What's the how important is that negative charge group on your polymer?
Oh, I mean, it's very important because it balances the positive charge on the ammonium here, right? So it has to be zwitterionic. The point is, I mean, this is a sulfonate group, and the point in having a sulfonate group is that first of all, it's quite let's say it's biocompatible. I mean, it's being used in many buffers. For instance, if you look at buffers like Hebes and so on, they all contain a sulfonate group. So it's something that is compatible with proteins, lipids, and so on. It's not reactive, and it is always negatively charged, yeah, because it has a pKa of, I think, something around 0 or even higher. So if you replace the carboxylate, you would probably have a pKa around 2 or 3 or so, right? Okay, I mean, probably for a biological experiment, it wouldn't make a difference as well, because you don't want to go down to those pH values either. We actually do have a series of polymers in which we have a carboxylate group instead of the sulfonate. But in all the experiments that we have done so far, the sulfonate performed better than the carboxylate.
Excellent, thank you. Any other questions? So I had one more question as well. So if you go back to the slide, you measure the size of the disc. I think that was like a slide 7 or 8. When you made a comparison. Yeah, this is slide. So the influence on lipid phase behavior, did you also measure the kinetics of solubilization?
We looked into the kinetics, yeah. Let me get this right. It was like for DMPC, for instance, the kinetics was slightly faster for SP digma as compared with digma, for SP SMA as compared with SMA. For negatively charged lipids like the MPG, for instance, the kinetics is much faster. So it seems to be due to the difference in Coulombic interactions, which also makes sense naively, right? So for zwitterionic lipids, we don't see a strong difference. We see some difference, but it's not very pronounced. But for anionic lipids, we see a huge difference. So for those lipids, the solubilization is much faster with SP digma and with SP SMA than with the unmodified polymers.
Thank you very much.
Can I ask one question?
Sure, please.
So I want to ask about the microfluid. So that is essentially a size determination experiment, if I understand correctly. Can you hear me? Sorry.
Sorry, can you say that again, please?
So like the microfluid, like the microtubes experiment.
This one here?
Yeah. So it's essentially for determination of the size of small, the particles. So what's the advantage of it, like comparing to other methods to determine the size of the particles?
Okay, the main advantage, I mean, there are a number of advantages. First of all, it is specific to the kind of particle that you want to look at, because you have to label the species of interest using a fluorophore, right? I mean, if you do dynamic light scattering, for instance, what you get is an average across all the different kinds of species in the incubation. That's not the case with MDS. So with MDS, you don't look at everything that you have. You just look at the species that you labeled with the fluorophore, because that's the only thing that you can actually see when you measure the concentrations at the end of the experiment. That's the major advantage. The second advantage is that it takes very little material. So because everything is microfluidics based, it takes just a few microliters of your solution. And since detection is through fluorescence, it's super sensitive, and that's essentially not limiting. Yeah, you can go into the sub-nanomolar range. So I think these are the two major advantages. First, specificity, you just see what you're interested in, and second, sample requirements. You can, for instance, measure in serum. If you have like a blood serum, if you take an extract of a cell culture, you can also use MDS. You couldn't use DLS because you would just get like very wild numbers that give you a gross average across everything that you have in this suspension. But with MDS, you can really specifically look at individual particles.
Yeah, thank you.
Dr. Sandra, Dr. Keller, I have one question for you. From our experience about this kind of polymer, you will get a small particle or you get a big batch. The size of the particle is small or big batch particle, and whether it can suitable for the cryo-EM or not.
Yeah, that's a good question. If this is suitable for cryo-EM or not. We haven't tried cryo-EM using those polymers. Yeah, I mean, everything I showed you was just negative stain EM. We haven't tried cryo-EM yet. We do have, our collaborators have preliminary data on cryo-EM that look very promising. But I'm a bit cautious. I wouldn't generalize this because, as far as I know, there is no good structure-activity correlation, right? I mean, there are a number of groups who've introduced polymers that should be particularly suited for cryo-EM, but I'm not sure. I mean, sometimes it works, sometimes it doesn't. And I think the major culprit is the heterogeneity in size that you get for different membranes, different lipid compositions. And some polymers tend to make more homogeneous nanodiscs than others, but in the end, it's not just the polymer, but it's also the proteins that you have in the nanodiscs, and it's also the lipids. Yeah? So, for instance, with glycodipma, so with the other polymer that I talked about last time, we saw that it works beautifully for mammalian cells. It works also quite good for bacterial cells, but it gave us very strange results for insect cells. Yeah? So then with this polymer, with these two polymers, collaborators got much better data with insects.
Yeah, because I missed some of your slide from the beginning. I just curious whether did you correct the size of the particle or not with the SB-SMA instead of the SB-DIPMA.
Yeah, I didn't show the data for SB-SMA because it's the same as SB-DIPMA. So we also get, I mean, with SB-SMA, the efficiency is slightly better than that of SMA. So the difference, so the improvement in solubilization efficiency is greater for SB-DIPMA than for SB-SMA, which is simply due to the fact that DIPMA is intrinsically less efficient than SMA. But the particles also end up at the hydrodynamic diameter of around 10 nanometers. So for SB-SMA, we also get nanodiscs. The efficiency is, I would say, to first approximation, is similar to that of SMA.
Okay, and how about the size of the particle if comparing with the SMA, the original one, without modification?
It's again about the same. So between SMA and SB SMA, we don't see a big difference. It's about the same.
Really? Because with the same, yeah, in our lab we use the same polymer, but we had a different result. But with SMA 2000, that is just an E. coli membrane, not human membrane. But we got a single particle. But in case of SB SMA, we got a big patch.
In the case of SB SMA,
yeah, SB SMA, we get a big patch particle instead of single particle.
Okay, are we speaking of the same SB SMA?
Yes, we have the same structure, but maybe the backbone, the ratio between the styrene and malic anhydride, it should be a little bit different. We use that is 1.5 to 1. I don't know in your case how much you use. That is 2:1 or 3:1. I don't know.
2 to 1. 2
yeah.
to 1.
But I think it could be not called the big problem about the size.
Yeah. No, I mean in our case we didn't observe this effect. It's
okay.
About the same for both.
Yeah, because usually for SB SMA, we got the big patch. So when we purify some protein, because if we get a big patch, a lot of the lipid there, so we can maintain the function very good. But for the single particle, we have the problem for the cryo-EM. Yeah. Oh, that all the thing. Thank you.
Go ahead.
So sorry about that. I have one question. From where normally we can purchase these polymers? I mean, SB SMA or SB DIPMA or yeah.
Sorry, I'm not sure if I got the question.
From where? How we can buy these polymers?
Okay, I hope that you will be soon able to buy them from Glycon Biochemicals. So the same company that is now really offering Glycodipma is now in the process of upscaling the synthesis of these polymers. So they're not available yet, but they should be available soon.
Thank you. And I also have one more question about the cell-free synthesis.
Yeah.
Why SMA has issues with that system, or why even DIPMA has issues with that?
I think this is because of the charge. Because what you need is magnesium, right? The translation machinery depends on magnesium. Without magnesium, the translation machinery cannot work. And too much magnesium is bad, too little magnesium is also bad. And if you now have the right magnesium concentration, you throw in lots of a polyanionic polymer, you will of course drastically change the concentration of free magnesium that is available to the translation machinery. So I think you simply screw up the magnesium concentration with those polyanionic polymers. And this is of course bad for SMA, and it's probably even worse for DIPMA, because DIPMA has even a higher charge density. And this is why we don't see those issues anymore with SB DIPMA and SB SMA, because there is no net charge.
Thank you very much.